For this lecture, you're going to need two colored pens. You can use a pencil or a pen, and one color. You could use a highlighter and a pen, but you need two things to write with that you can tell the difference between. So we're going to look at the first page. We're going to title it Asexual Reproduction. Asexual reproduction is any time you reproduce using only one organism. So we're going to write that organism. Unlike us, where it requires two parents, this is where you only need one parent. So the first example is with bacteria. All bacteria do is that they copy their DNA and they split in half. This is actually called binary fission. Splits in half. The daughter cells, the daughter prokaryotes, are identical to parent. That's the most common kind of asexual reproduction. All the bacteria in the world reproduce that way. There's some interesting details, but we're not going to go into them. Some plants can reproduce themselves asexually. So for instance, strawberries can send out little runners and make copies of themselves. Some animals, not very many animals, very infrequently. This is called a hydra. This hydra can reproduce itself using asexual reproduction. It is not called binary fission. Binary fission is just for prokaryotes. And then lastly is this one that we've seen a few times, mitosis. So it's how you can reproduce eukaryotic cells. In our body, it's how we reproduce most of our cells. So we're going to turn the page. For me, it's just lifting up the page. I'm going to draw you a picture. I'm going to draw you a picture of this guy named Fred. He's from the Fred species. I don't know why. Yours does not need to be beautiful. So this is Fred. The only point is he's a really weird species that doesn't exist. We're going to draw Fred's karyotype. Fred's karyotype. This is where you need your two colored pens. Fred's karyotype. Make this one big, medium, small. Big, medium, small. He has 1, 2, 3. That is it. That is his whole karyotype. So remember, N is number of chromosomes in one set. You saw that last night in the video, and it's up on our board. Right now, when I say pause the video for a second and see if you can figure out for Fred, N equals what? So go ahead and pause, see if you can figure that out. You can check your notes. All right, so for Fred, one set of DNA. He has two sets. If I look at all the red chromosomes, there are 3 of them, so N equals 3 for Fred. That means in each body cell of Fred's, how many chromosomes does he have? You can look right here. 6 chromosomes per somatic cell. Somatic cells are all cells in your body except for the gametes, except for the sperm and eggs. I'm going to draw some pictures over here because I never showed this to you before, but I'm going to show you what it looks like when a chromosome goes through S phase. So up on the board I have 2 homologous chromosomes. So I'm going to draw Fred's chromosome 1s in a much prettier way. One, 2. So these are chromosome 1. They are my homologous chromosomes. And they are unduplicated. So if you remember back to mitosis, they haven't gone through S phase. They haven't made copies of themselves. If they went through S phase, synthesis phase, I would actually draw them like this. So here's my original chromosome 1. And my second chromosome 1 is attached. Remember how we called this little spot right here the centromere? They're attached right at the centromere. And then the same thing for my homologous chromosome 1. I now have a duplicated copy. So what we say is these are unduplicated homologous chromosomes. And these are duplicated homologous chromosomes. I still have 2 chromosomes here. I have 2 chromosomes here. The difference is these are unduplicated, these are duplicated. So we're going to draw in a quick picture. I'm not always going to draw my chromosomes this pretty. So if I were going to draw Fred in here, I'm going to just draw, these are unduplicated chromosomes, I'm just going to draw a line instead of or a thicker shape. There are his 3 pairs of chromosomes. This is a somatic cell. It is a diploid cell. There are 2 sets of chromosomes. 3 plus 3. Also known as 2N. I have 2 sets of 3 chromosomes. Then Fred might go through S phase. His chromosomes would look like this. They're attached at the centromere. And he still has 2 sets of chromosomes. They're just duplicated. So weirdly enough, this is still diploid. It's 2 duplicated sets of chromosomes. And then, if he goes through mitosis, which we'll talk about in more detail, he will have, I should have drawn these in half. These will basically split, and he will have 2 identical daughter cells. Oh shoot. There we go. 1 copy will go into each cell. So he still has exactly the same genome, exactly the same as he did before. These are 2 identical daughter cells. That is it for the notes for right there. Pause and go online and take the exit ticket before you come back to the next video.